in summary all the 4 lectures on systems biology. That is like here systems biology, the general topics which I will be covering, and the typical questions of systems biology. So before I move it on the topic, I will request you all to subscribe our channel and also make the comments. If you want to have some comments, please make sure to have some comments, question answers, so that I will try my best to answer your questions. So here in general topics, I will be presenting the networks of gene regulation and metabolism, signaling cascades in a cell, paths from the genome to cellular function, multicellular systems, and the typical questions of system biology, that is an organism viable under specific perturbations, on which level of organization a particular function located, what are the effective system boundaries. Now, so in system biology, you will be learning, which I have already discussed in the last 3 4 lectures, that networks of genes, proteins, and metabolic or metabolites. So here you can see one basic example of the network as a graph. Here you can see the nodes or vertices in red color, which could be genes, metabolites, or proteins, and then links or edges, which are regulation, reactions, or physical bindings. So here, systems biology, what are the main challenges? Genes are distributed along the genome. Genes regulate the other genes. Genes produce enzymes which catalyze metabolic reactions. And then examples you can see, the E. coli has about 4,000 genes, and then about 1,200 metabolic reactions are present or possibly. Entangled 3D structure of the genome. Now, what is the challenge? The structure is highly complex, whether it is a small organism or bigger organism. So here you can see on the left side, that is metabolic network example or metabolic process, that is metabolism. Then on the right side, it represents the gene regulation process, which appears to be highly complex. And that is why you need to enter, instead of going to biological experiments, you need to enter into systems biology. Now, if you want to expand certain part of the network, then this network could look like, here you can see the enzymes E1, E2, E3, E4, and so on. And then the structures are quite like diverse. So the highly heterogeneous structures are present, which is highly difficult to unravel or understand with your open eyes or manually. Now, in order to understand the network structure, you need to go for the how the structures perform dynamic tasks. Then, what is the role of biomass and how the distribution of metabolic fluxes are possible, response of the system to metabolic perturbations. You need to understand about 85% accuracy in predicting lethal mutations are possible so far. Steady state theory, no complex dynamics. Why can a steady state theory describe metabolism so successfully? When do more complex forms of dynamics matter? Now, comparison of expression changes between different experimental conditions, and then comparison with the gene regulatory networks. So you can try to understand with the experimental data available, or you are going to generate, and then build the theoretical gene regulatory network, and finally go for the effective networks, which you can see in the red color here. Now, the challenges could be, for example, wild type network, where there is no mutation, no perturbation. Then mutated network is here. Then another mutant network is here. So the effective transcription regulatory network is more densely connected for the mutants compared to the wild type. Perturbing the network by mutation enhances the network uses. So the take-home message here is you need to understand when there is perturbation in the network, they are more densely connected for the mutants. And with respect to the wild type, where there is no perturbation. Now, perturbing the network by mutation enhances the network usage. So the challenge of null models in system biology, the link between topology and dynamics, you need to understand the link between topology and dynamics for the complex systems. You need to also understand, for example, brain activity, epidemic spreading of the disease in social networks, transport logistics, organization of metabolic fluxes. Then the imprint of evolution in biochemical networks, understand the gene regulation, where you need to understand the digital functions versus continuous stochastic biochemical implementations, predicting biological patterns, and then decision process or distributed computation in biology. Now, what are the networks which you are supposed to see here? As I mentioned here, there will be two types of links between the topology and dynamics. For example, here you can see the links, and then there are quite possible between these networks that you need to go for topological analysis, the behavior of the network, the connectivity of the network, the adjacency, and many more things which you can analyze for a given network. Now, you have already seen the network, how they mutated, how the wild type networks look like. Now, what in general is the challenge? What are the promising point? Here it says in one of the essay paper, mathematics is biology's next microscope, only better. Biology is in mathematics next physics, only better. So this you can also read the details. So what it says about systems biology role and what are the future challenges. So a popular notion of complex system is of very large numbers of simple and identical elements interacting to produce complex behaviors. In biology, large number of functionally diverse and frequently multifunctional sets of elements interact selectively and non-linearly to produce coherent rather than complex behaviors. What are the responsibilities? Balance between the two types of complexities, close contact between theory and experiment, which you need to establish. Analysis of high throughput data and mathematical modeling of biological process is required. Strategies, abstraction of complex biological process as graphs, steady state models of the metabolism of a full organism, detailed models of a specific pathways. Then, as I mentioned, the general topics, networks of gene regulation and metabolism, signaling cascades, paths from the genome and cellular function, multicellular function, and the typical questions. So now we come to the levels of cellular organizations. I have also presented in the previous class that genomics, transcriptomics, proteomics, metabolomics, glycomics, and lipidomics. And finally, you can move towards the phenomics. So, on what basis? Here you need to understand the interactions, the control and coordinations at different levels. For example, genomics, transcriptomics, proteomics, metabolomics, glycomics, lipidomics, and localizomics. So here you need to understand what are the association or what kind of linkage exists between these components. So finally, what will be the functional states that could be fluxomics or phenomics? And in between, you have these kind of things which you need to perform by using systems biology. Now, if you want to understand the roles of components of the networks, here in case of metabolic networks, nodes are metabolites. In protein interaction networks, nodes are proteins. In gene regulating networks, nodes are genes. In protein contact networks, nodes are amino acids. And finally, food webs, here the species are the nodes. Links are also different, like reactions and enzymes in metabolites. And then physical binding in protein interaction, transcription factors in gene regulatory networks, a spatial proximity in protein contacts, and then in food web, it is predation. What are the signals? exchanged groups or atoms, logical operation or the inhibition activations. Then biomass in food web. In topology, in how you can see enzyme inventory, list of chemical reaction, Y2H here in protein interaction networks, binding sites, expression data, generality networks, protein structure in protein contact network, dedicated experiments on small ecosystems. And in terms of dynamics data, metabolomics data, metabolic fluxes, time resolved microarray data, generality networks, or these days NGS also. Diverse and linked to topologies in food webs. Now, metabolic network examples you can understand from Paulson papers and books. This is the book on metabolic networks or metabolic system biology from Paulson. Then here you can see how the metabolic reactions may look like and how the metabolic network will look like. They are presented in parallel. Now, here protein interaction networks, you can see two proteins are interacting with each other. So, irrespective of the name of the proteins, you need to understand more than one protein could interact and then they could regulate the biological functions. Then here you can see Y2H and then reporter gene and some proteins, genes, some other genes also possibly available. You can read some of the research or review paper on protein interaction networks. Then, if you want to go for complex protein interaction network, it may look like such huge, but this is even a smaller network. It is much bigger and complex in higher organisms. Now, you can see the protein networks before and after duplication. So these are the kind of perturbation which you can allow. Now you can move to gene regulatory networks. So you need to understand the location of the genes on the chromosomes and chromosomal indexes or positions. So a start and end positions on each chromosome or on different chromosomes. Then here you can see the gene regulatory networks, how they look like in this analysis. Now, if you want to go for protein contact network, you can see here, these are the amino acids as the nodes. And in overall, the logic remains same irrespective of the type of network. So you need to perform the network topological analysis. Then it comes to food wave. You can see here. So what, by the way, if some people can ask what is the difference between protein interaction and protein contact network. Here you can easily understand the PCN or protein contact network is a graph-based representation of a 3D protein structure where amino acid residues are nodes and non-covalent interactions between them are edges. This network analysis reveals insight into protein stability, function, allostery, and dynamics by identifying key residues, modular substructures, and communication pathways within the proteins. Now it comes to food web. Similarly, the food web is a complex network that shows the flow of energy and nutrients through an ecosystem, illustrating the intricate feeding relationship between different species. It is made of many interconnected and overlapping food chains, which provide a more realistic model than a single food chain to understand the complex interactions of organisms and how energy moves from producers to consumers and decomposers. This is what you could learn from food waves. Now there are some examples for drug development. This I have given some realistic examples in the past lectures also, but here you can understand how the system biology is playing crucial role in understanding the drug development. So here, for example, from Wikipedia, you can see the EGFR as one of the example, then P53 as another example. Then if you want to unravel it, then there are several papers, research papers, where it has been potentially shown that system biology could perform a dynamic role in drug development and drug discovery. So here EGFR, epidermal growth factor receptor, mediates increased cell proliferation, migration, and aggregation in esophageal keratinocytes in vitro and in vivo studies. Then there is another paper as a review paper, a comprehensive pathway map of EGFR signaling. So if you will read these papers, you can easily understand this is the another map of for representing the EGFR complex pathways, and this you can also learn the different components, downstream signaling molecules, and many more things. Now, the symbols for that complex networks are the details here. You can see, for example, protein is represented by another structure, receptor is represented by another block or box, ion channel is represented by another symbol. So these are the things which you need to. Then it comes to reactions. So how will you identify the different types of reactions, state transition, known transition, unknown transition, transport, transcription, different things you can understand. Then it comes to some more things which you can also understand by reading the review and research paper. This is another network for representing the complex EGFR signaling pathways. If you understand this, the down here are written is basically cell-fate decision, apoptosis, proliferation, or what kind of role this EGFR could play as an overall, as a network or within the by using this complex network. Now you can go for another example, that is TP53. So it is basically 494 amino acid protein. It is called guardian of the genome, tumor suppressor due to its function in the network of transcription factors, activation, inhibition, degradation by standard processes. Target of P53 have similar regulatory mechanisms. None of these ingredients alone would function as a tumor suppressor, on which systemic level is a particular biological function situated. So here you need to understand why this TP53 or P53 is so important, and what are the multifaceted role of TP53 in different kinds of cancers and different human diseases. So here, as a network, if you want to see in complex way how this is TP53 is regulating DNA damage, which is very important in cancer, or in, you can say, controlling the cancer process, or in association with cancer, different types of cancers. It's not with one. Now it comes to TP53 target genes locations on different chromosomes. These genes highlighted are basically indirectly directly interacting with P53, and they are located on almost all the chromosomes except 4 or 5 chromosomes. For example, this chromosome number 4 has no TP53 here, and then, but this is the old slide. You can also look for the newer study where you can see the location of TP53 even on chromosome number 4 or maybe some other chromosomes. So you can now understand how impactful role TP53 could play as a whole. Now you can see the summarized map of the T53 or TP53 network, which is here like P21, CDK2, then MDM2, ARF. These three basically they are quite potentially associated with, directly associated with cell fate decisions. Now, with the challenges, you need to understand their structures. For example, in metabolism, the network structure is slightly different. Gene regulation is quite different. But if you want to understand more, you need to understand the smaller or subunits of the networks, that is also called network motifs, which I will be discussing in other classes. But here you need to divide and the networks into groups and understand, or maybe you can call it as clustering. Perform clustering and try to understand the clusters, how they look like. Then it comes to another example, which you can see. There could be many more examples for understanding and unraveling the network structure and their role in terms of systems biology or in terms of biological functions. So that's overall what I wanted to say today. And today I would like to say thank you for all those people who have subscribed and helped me to reach or cross 1,000 subscribers. And please continue to subscribe and like and make comments and visit our channel. Thank you very much. And I highly appreciate all the people who are watching my channel. Thank you all.